All right. Hi, my name is Katrina Ashley. I am in Professor Gengshi Liu's Microbiology for Allied Health class. And my project is about the bacteria of local compost. And so I'm going to share today some of the exploration that I did into my own personal compost pile. It just so happens that this is International Compost Awareness Week, which I promise you I did not plan. I just found out about it yesterday, but it fits pretty well. It's my first slide here. Home gardening has seen a substantial increase in popularity in the last 2 years. The original spike was because of the COVID-19 lockdowns. More time at home meant that a lot of people were looking for new activities. I know I picked up probably a dozen hobbies, and gardening was one of those activities for a lot of people. One study found that the pandemic caused at least an 8% increase in sales in the gardening industry. And since then, supply chain issues and a fear of empty grocery store shelves and the rising cost of food have all helped to encourage people to grow their own food. But anybody who has gardened can tell you that it is surprisingly expensive. You might start by grabbing those deceptively cheap dollar packets of seeds at Walmart, but then when you're checking out an hour later with $100 of soil, fertilizer, and tomato cages, it doesn't feel so affordable anymore. But thankfully, compost is a high value, low cost source of nutrients for plants. I had to throw in a picture here in the top left of my own potato plants this year, which are growing very happily in soil mix that's about a third compost. And almost anybody can compost at home for free. At its simplest, you just throw produce scraps, eggshells, cardboard, yard waste into a pile or a container. You stir it now and then. And within a few months, you have what gardeners refer to as black gold, which is the rich soil-like product of the decomposition of organic matter. But what turns those scraps into compost, that decomposition is a result of a massive network of micro and macroorganisms. Insects, worms, fungi, bacteria all work together in that process, but the most productive are the bacteria. So for my project, I sampled my home compost pile and I investigated the bacteria from it. This bottom picture here is my compost pile. You can see a few different kinds of materials there, some straw and pine shavings, the watermelon that my mom's chickens just finished enjoying. There's another melon further along in the decomposition process, and then some of that darker, more mature compost, which is what I sampled for the project. So I hypothesized that I would find thousands of bacteria and that they would thrive in aerobic environments with oxygen present, and that they would produce a wide variety of enzymes in order to break down a wide variety of materials. So my first step was taking my compost sample to the lab and diluting it in order to estimate the bacterial count. So I put 0.5 gram of compost into a 5 milliliter tube of nutrient broth, mixed it up really well, and then progressively diluted it 4 more times and incubated a spread plate for each dilution, which you can see here. I was looking for a plate with few enough colonies to count accurately, but enough to be a good representation of the original sample. You can see plates 1 through 3 have just too many colonies to count, and plate 5 only has a couple, so it's not representative. So I counted plate 4 and I came up with 33 colonies. So my estimated colony count, when you do the math, works out to about 330,000 colonies per gram of compost. So my next step was to choose a few colonies to focus on. I chose 3 interesting colonies and I used streak plate technique to isolate them and performed a gram stain on each, which allows us to see the type of cell wall and helps us to categorize the bacteria. So you can see the 3 colonies that I chose here. The first one was the only pigmented colony on all of the plates. It's this kind of distinctive orange color. The second was more typical of most of the colonies, a colorless, mostly round growth. And the third was colorless, but it had this really interesting, hairy, swirling growth pattern. And so when I performed the gram stain on each sample, I found that they were all bacillus or rod-shaped bacteria. And samples 1 and 2 stained pink, indicating gram-negative bacteria, and sample 3 stained purple, indicating gram-positive. And so I set up about a dozen tests for each of my samples. So instead of going through them all, I'm just going to highlight a couple of things that I found interesting. So first, I'd hypothesized that I would see a lot of different enzyme activity, and I didn't see nearly as much variety in each bacteria as I expected. A lot of my enzyme tests were negative. And then this top right picture, my sample number 3 was motile, as you can see in the cloudiness through the tube there. It just spread all throughout the tube. And one really unusual thing that I noticed is that it also liquefied the sim media at the bottom of the tube and produced a gas bubble, which is not something I had seen before with that media. And then all three samples, oh, my dog's not happy, fermented glucose and either lactose or sucrose or both. It's these yellow tubes at the bottom. And then this bottom right image here is the starch hydrolysis test. I included this picture because it was the most visually striking to me of this project, and it really kind of made me appreciate the strange beauty of bacteria. The orange pigmented bacteria has hydrolyzed or broken down the starch around it, as you can see by that clear halo. And the plate at the bottom is the third sample. And even though all three cultures were incubated for the same amount of time, you can see how much faster the sample spread. It takes up almost the whole plate. And you can see these really interesting edges of the bacteria against the dark starch and iodine media. It actually reminded me of like a map of a coastline where like the growth is the coastline and the media is a body of water. It reminded me of the Chesapeake Bay a little bit. And I found it really fascinating. I really loved that reaction. My full chart of results is on this slide, but it's quite a bit to read. So with these test results, I dug into researching these bacteria to attempt to identify them. And I knew I wasn't likely to be able to make specific IDs, but I wanted to get as close as possible. So I started with Bergey's Manual of Determinative Bacteriology, which made it really simple to compare characteristics between family, genus, and species. So sample 1 has that distinctive pigmentation, that orange color, which matched well with the genus Flavobacterium, which are gram-negative aerobic rods. And most other characteristics fit that genus, but the catalase test, that test for the enzyme that breaks down hydrogen peroxide, was negative for my sample and is typically positive in Flavobacterium, so that conflicted. But no other genus was as close a match as Flavobacterium. So I started to suspect maybe my test was flawed, but when I researched online, I found at least 3 Flavobacterium species, F. cumensi, F. urolithicum, and F. alvei, that have been identified in the last few years that are all catalase negative also. So that suggests to me that there may be catalase negative species of Flavobacterium that aren't included in my 20-year-old edition of Bergey's Manual. But it's still possible my test was flawed or that the bacteria belongs to a different genus I wasn't able to identify. Sample 2 had the least distinctive morphology of the 3, and the characteristics that fit the family Enterobacteriaceae, but I was unable to identify it any further. I didn't find any species of the very many listed that fit perfectly with the results from my culture. But Enterobacteriaceae are common in soil, so I feel comfortable identifying it as a species of that family, and I would probably need to run some more specific tests to narrow it down any further. And then sample number 3 aligned with the genus Bacillus. Bergey's Manual doesn't differentiate any further with this genus, but I spent some more time on research, and because of the unusual rhizoid colony morphology, that hairy swirly growth that made this one so interesting, I was able to make a likely identification. So this bacteria is likely to be Bacillus mycoides, which is an aerobic mesophilic bacterium that's very common in soil and water. The myco in the species name refers to the fungus-like growth pattern. And here on the slide, you can see my culture on the right, and then on the left are 2 samples of Bacillus mycoides from a study that investigated the direction of the swirl pattern in the growth. So it's a pretty good comparison, I think. So in the end, part of my hypothesis was confirmed and part wasn't. The bacteria that I tested didn't utilize as many enzymes or energy sources as I expected it to, which leads me to believe that the diversity in the bacterial ability in compost is probably due more to the variety of bacteria than the varying abilities of each species. But I would have to do more research to confirm that hypothesis. I did confirm my hypothesis that I would find thousands of bacteria in the compost. 1 gram of compost isn't even enough to fill a teaspoon, but it contained over 300,000 bacteria, so it's absolutely brimming with activity. And in fact, I can be certain that the actual count is much higher than that because many other compost bacteria thrive at lower or higher temperatures than I used and might not grow on the specific media that I used. And that count also doesn't include fungi or actinomycetes, which are a special bacteria that are similar to fungi that give soil its distinctive smell. So the flourishing population that I counted suggests a vast and powerful network of decomposers in the compost. Bacteria often come with negative connotations, but we know that the majority of bacteria are beneficial or harmless, and the same is true here. Bacteria are essential to the production of compost, so these little microscopic powerhouses can help to provide an effective, inexpensive source of plant nutrition for home gardeners. And I can attest that my plants love the product of the bacteria's work, and I am glad to have them on my team in my quest for homegrown produce. And then finally, I'd just like to thank FCC and the Honors College for the chance to study independently and learn more, and for holding this forum for us to share our projects. And I also want to thank Gengshi for encouraging me to pursue this project and for your support along the way. It's been a privilege to take your class, and I've really discovered a love of this kind of research that I didn't know I had before this semester. And thank you to you all for listening. So if anybody has any questions, go for it.